So we've been studying smoldering myeloma. Smoldering myeloma, as you all know, is a space of immense research interrogation, and it's a really interesting space because as of today we still say that myeloma is incurable. And if you start thinking about, well, where can we cure myeloma? You have to cure it at probably its earliest stages. The good news in myeloma is you have this precursor disease state, which is smoldering myeloma. The other bit of good news is we also know that who are the ones who are going to progress within the next 5 years. We've done some kind of risk stratification. It's not perfect, and we're still trying to modify that and work on that to become better at prognosticating who are the ones who are going to actively develop myeloma. But until then, what we did here was use sort of a minimalistic approach, because if you go on to clinicaltrials.gov, you're going to see the whole entire spectrum. Murray B. Mateos is doing kind of the, KRD with DARA, with consolidation, with transplant for smoldering myeloma, and we at the other end decided to use an immune-based approach. And in this case, what we did was vaccinate patients, and this is a multi-peptide vaccine. It's a vaccine which recognizes several different proteins. It recognizes CD38, which is pretty much present on all myeloma cells. It recognizes CS1, which is SLAMF7, which is the same protein recognized by elotuzumab, and it also has two forms of XBP1, the total form and the spliced form, which is really important in multiple myeloma biology. So these are 4 peptides put together, and we've just published this, as you mentioned, in JAMA Oncology, and what we've shown is our experience with the first 22 patients. These were in 2 cohorts. The first cohort was vaccine alone, the second cohort was vaccine with lenalidomide, and the vaccine is given 6 doses of the vaccine given at 2-week intervals over a 3-month period, and then we checked their immunogenicity at month 3, month 6, and a year out. And what we saw in this trial was when they got the vaccine alone, it was well tolerated. We did see some immune responses, including the presence of memory T cells, which to me is actually quite remarkable. If you have tetramer positive memory T cells, it suggests to me that the vaccine is waking up some of those T cells and is creating a memory in those T cells to hopefully then prevent the resurgence of myeloma. And what we then found in the follow-up cohort where we added lenalidomide is this immune response was augmented even further. So the amount of CTLs, or the cytotoxic T lymphocytes to all these different peptides that I've talked about, were increased in patients who got lenalidomide. The memory T cells were also increased, and all we've studied right now is their immune response. Obviously, we have to follow these patients long enough to see whether or not we can prevent their progression. We're already doing the next rendition of the study, which is the next cohort, wherein we are doing lenalidomide with an HDAC inhibitor. This is an HDAC sort of 6 selective inhibitor, where we've shown that exposure to that HDAC6 cytarinostat in this case augments certain features within the T cells. It augments co-stimulatory domains, it augments NK cell activity, it decreases T regulatory activity, which is bad for immune responses. So we're going to combine this with lenalidomide and the vaccine and see whether or not we can even do better than what we are doing. So it's exciting, but it's really early data. But the good news is, this would be a preventive strategy to prevent the actual development of multiple myeloma.